Every hour, one man dies from prostate cancer. That's more than 11,000 men every year. Despite efforts to screen and diagnose men early, 29,000 prostate cancer-related deaths was recorded in 2013. 47,000 men across the UK and 3,000 across Scotland alone was diagnosed. Healthcare professionals will look at the levels of prostate-specific antigen, or PSA, to provide a positive diagnosis. However, PSA is not very specific. Although it's known to be elevated in prostate cancers, the levels in the blood can be misleading. Some men have naturally higher levels of PSA, which leads to overdiagnosis and overtreatment. On the other hand, some men will not be diagnosed at all. Their fast-growing cancer does not show the typical levels of elevated PSA. So this is where our lab comes in. Our lab has revealed that the enzyme PDE47, or 47 for short, could be an alternative to PSA. So PDE47 significantly increases in expression at the initial stages of disease. However, as the disease progresses to the more incurable state, its expression completely decreases to an undetectable state. And this is where my thesis comes in, which can be described in two parts. So firstly, I will be further looking at the possibility of making 47 a new biomarker for prostate cancer diagnosis. I will be looking at the genetic profile of this enzyme in human disease samples, as we believe, as well as my founding body, Philips Diagnostics, that will be a better tool to predict relapse as well as disease progression. Secondly, I will be looking at the molecular events that leads to the decrease of this enzyme in human samples. If we can understand why 47 decreases, then we can develop drugs around 47 as well as any interactive proteins such as the androgen receptor to prevent the disease from progressing in the first place. PDE47 has the potential to be a better biomarker to diagnose prostate cancer, as well as being a new therapeutic target for drug therapy. Thank you.